Great. Well, first of all, I don't know if you can hear me all the way back there in hall 100. Thank you very much for coming here. I want to thank Professor Vincent very much for this conference. I think I speak for all of us on how fantastic it was. Yeah, sure.
It's recorded, so we have to get it out.
I see, okay. I was asked to give this lecture on how cells die. It's like giving a lecture on immunology for 15 minutes. So it's a very complicated subject, much more complicated than even I had anticipated. So it will be a large overview on the subject of death. Now, I'm a physiologist, la science de la vie. So to talk about death is counterintuitive for me and very difficult. But it reminds me of this wonderful saying by Epicurus, the art of living is well, art of living well and the art of dying well are one. So if we want to understand how to live, we must get insight into how cells die. Because of course it is the underlying cause of organ failure ultimately. And there are very many pathways. Now, the classic type of cell death, which we all know, I think, from our physiology books, really is apoptosis, autophagy, and necrosis. Those were the three main ones. Apoptosis is programmed cell death. Autophagy is cleaning up the inside of the cells. And necrosis is just unprogrammed cell death. So if we look at what this means, the necrosis and the apoptosis, apoptosis is a lot more controlled type of a programmed cell death. It usually involves single cells and it requires ATP. It's an energy-dependent process. And there is little release of cell content, and the most important aspect of apoptosis, it's not immunogenic. You do not get an inflammatory response. Conversely, necrosis usually occurs in a large number of cells together as a result of some form of an injury. It can be radiation, it can be hypoxemia or ischemia reperfusion, etc. The cell contents are released, and necrosis has got a strong inflammatory response. Well, we thought, well, okay, let's look at those three. No, unfortunately, history is very unkind to us, because since the discovery of apoptosis, or the description of apoptosis, actually already described by Hippocrates many, many hundreds of years ago, by Kerr and colleagues, various different types of cell deaths have been included over the years. And I'm not going to go into each one of these, but I am going to tell you, to show you the scene, as it were, of cell death. And in 2018, there was a consensus meeting to try and bring some order into all of these different mechanisms involved in cell death. We are talking about really regulated cell death. So there are some issues relating to pathways, internal, external pathways, intrinsic, extrinsic pathways, ultimately resulting in cell rupture and cell death. So in 2018, a committee on cell death, they have committees, they even have journals on cell death, got together and they came up with a nomenclature of cell death. And my God, look at all of these. Erythrocytes, endothelial cell death, Eryptosis, etc., etc., mitotic cell death. And they are really quite distinct from each other. So the bottom line story here is that there are many, many pathways to die. Let's look at some of them. So here we've got a number of the type of cell deaths which have been described. They usually have been categorized according to their morphological characteristics associated with cell death in combination with the biochemical pathways resulting in the pathways. So the morphological features really range from a fully necrotic type of cell death to fully apoptotic, which is the most silent way of dying, as it were, and ranging also from anti-inflammatory to pro-inflammatory. So a lot of these processes either are used in the body to instigate an inflammatory response, and some the opposite. And here we've got some of them: immunogenic cell death, lysosomal dependent cell death. So, of course, a lot of the cells in our body have got a lot of cytotoxic compounds inside their membranes, which are used for phagocytosis, intracellular killing, etc. These are oxygen radicals, but also lysosomal content. And these are involved either in a controlled or an uncontrolled manner in instigating cell death. Okay, here we go, guys. There's intrinsic apoptosis and extrinsic apoptosis. So this is a signal coming from outside of the cell, inducing the cell to commit suicide, or internal signals from inside. Then there's the mitochondrial pore transition, which is basically pores which are punched into the mitochondria, by which the mitochondria loses its membrane potential and the mitochondria basically become energy depleted. It's also one of the most important pathways. We have necroptosis, which is like necrosis, except it's programmed, and it elicits an immune response. Ferroptosis is specifically associated with the iron metabolism in the cell and is associated with oxygen radical and the injury that oxygen radical forms. Pyroptosis is a apoptosis type of process, but resulting in the release of cytokines and eliciting an immune response. So you see all of these different ones have got different ones. Then we've got the lysosomal dependent cell death, in which lysosomes, instead of involving in the clearance of pathogens, like intracellular killing of Salmonella typhimurium, for example, by monocytes or macrophages, are actually having a sort of an autoimmune type of a function in which certain perturbations results in an abnormal intracellular hemostasis resulting in cell death. The final one, and I will end my talk with a discussion, is cellular senescence. And I think this one is probably the one which certainly is most relevant to intensive care medicine, because what a cellular senescence basically means is that a cell all of a sudden is not really functioning well. It is still metabolic active. It is still using all sorts of compounds required for cell survival, except they do not contribute to cell function. This is in organs, for example, that have got a large number of fibroblasts. So fibrosis in the heart, in the liver, and in the kidney contribute to organ dysfunction because they are taking up space by functional cells, and they are called zombie cells, actually. And in acute kidney injury, which I will show you, it's the best described, because what you have is a cell cycle after acute kidney injury. You've got a cell cycle which results in epithelial cells, again, contributing to the barrier function and the sodium reabsorption in the kidney. However, it does not complete the complete cycle, and it arrests in the cell cycle, and it differentiates out into fibroblasts. So the kidney starts getting a lot of fibroblasts, a lot of cells which have no business there, and it is one thought to be a main contributor to chronic kidney disease. We can go on and on. So if we now look at all these different types of cell deaths, they are basically all can be categorized into different types of morphological features. So these are usually microscopic anatomical issues. So here we've got membrane rupture, blebs, for example, formation in the phospholipid membranes. We've got autophagosomes. So as you know, phagosomes are usually associated with clearing of extracellular pathogens, but intracellular autophagosomes are actually phagosomes which are related to intracellular organelles inside the cells, and so on and so forth. So let's focus a bit more on the sepsis part of the story. And the sepsis part of the story really is very complicated because when we talk about normal physiology, all of these processes related to cell death are usually associated with adaptive mechanisms that the body has to deal with issues related to, for example, infection or organ injury or some trauma, etc. And they are usually associated with wound repair, etc., clearing of debris. And these are garbage collectors, if you like, of the body. Now, in sepsis or in systemic inflammation, everything goes wrong. Not only our whole homeostasis doesn't work anymore, but also the regulation, the programmed regulation of cell death is no longer working. So apoptosis is a good example, which is usually a good thing which happens in the body, but now it gets frozen in certain processes and we are getting an abnormal regulation of cell death. And also, what's very important is that cell death pathways are one of the major contributors to an abnormal functioning immune system. And that's what this slide shows very nicely. So we have here in sepsis the innate immune system with the dendritic cells, the macrophages, neutrophils, and natural killer cells. And here we've got the adaptive immune system with the T cells. And if we see what all happens really during sepsis in these cells, we see that practically all of them, excuse me, start getting increased amounts of oxidative stress, cytokines, etc., all the things we talk about here in Brussels. And almost all of them really finalize in a pathway related to apoptosis, with the exception of the regulatory T cells. So apoptosis is really, and the time dependency of apoptosis is one of the hallmarks of the, first of all, the activation of the immune cells, and after that, the exhaustion of the immune cells, in which immune cells are not functioning well. And we always know that there's this two cycle going in, the cytokine storm, followed by an exhaustive, depleted immune system. And cell death, immune cell death, is a very, very important component of that. So what is apoptosis exactly, and how does it work? We could spend a whole day on apoptosis, but basically what apoptosis does is it activates the caspases. And caspases are a number of a chain reaction, as it were. Normally they are inactive in proenzymes. Once they are activated by pathogens or by also different sorts of compounds, they will go into, which is a bit like the complement system, caspase 2, 8, 9, 10. These are the initiator caspases, executioner caspases, and once they produce a chain reaction, they activate executioner caspases. And these executioner caspases are caspases which can do very bad things. In fact, what they do is they can cleave up to 600 cellular components. So caspases are like a super, say, oxidative stress. So what happens then is that these caspases start to cause proteolytic cascades, resulting in chromatin condensation, DNA fragmentation, intracellular substrates, they all get chopped into little pieces, and ultimately resulting in cell death. But it is a programmed cell death. So the second part of death signals or death initiators is, of course, oxidative stress. And again, oxidative stress, too, is a very complicated and very extensive methodologies, but the major source of oxidative stress and producing of reactive oxygen species are the mitochondria. So if we have any problem relating to mitochondria, we have the PAMPs and the DAMPs, of course, which are inducing intracellular activation of various pathways. And we know that there are various enzyme systems like inducible nitric oxide synthase, but also reactive oxygen species related to the mitochondria. And these have all sorts of effects on the respiratory chain and on the membrane potential of the mitochondria, which are all important functional aspects of that. I will show you that in a second. So getting back to apoptosis, why is apoptosis not immunogenic? Because it's a very beautifully programmed cycle. Here we have a normal cell. First the cell shrinks and we get all of the chromatin condensing. The organelle starts to disintegrate, and if you look under the microscope, you can see little blebs forming in the membranes. It then starts to form what are called apoptotic bodies, which are then internalized by the macrophages and cleared by intracellular lysosomes. And that is the reason, because the macrophages are actually clearing all of these apoptotic bodies, you do not get an immunogenic response. Beautiful system. So you basically have got two pathways for apoptosis. You've got the extrinsic pathway, which attaches to the death receptor, which is a very scary receptor. And you've got the intrinsic pathway, which is very clearly associated with mitochondrial issues. They've got different caspase pathways, eventually ending up to caspase 3 to 7, resulting in substrate cleavage and eventual cell death. That is apoptosis. Now, some of these caspase subgroups will initiate specific types of cell death, which have specific phenotypes. The best one described for that is so-called pyroptosis, and it is related to the activation of caspase 1. And what caspase 1 does actually, it does a whole bunch of very nasty things inside the cell. It starts to cleave DNA. It forms pores in the cell membrane, enormously large pores, a bit the same as complement 9 pores. They're like 1 to 3 nanometers in size. You can see them on electron microscopy. It causes cell swelling and release of intracellular activations of cytokines. So that's why pyroptosis is very, very immunogenic and causes activation. And all of this results, finally, of course, these pores cause a depolarization of the cell. It loses its ability to generate energy, and resulting again in cell death. Then we have necroptosis. Necroptosis at this moment is very, very hot in the tumor literature, because necroptosis is really a programmed form of necrosis. So we heard that necrosis is unprogrammed. It occurs spontaneously as a result of an injury, but necroptosis is programmed necrosis, and it also is involved in a spilling of the contents of cells into the environment, resulting in activation of endothelial cells, macrophages, etc. So then we have autophagy. So this is self-eating. It is cannibalism, actually. So basically what happens in autophagy, they also can be induced by extrinsic or internal factors, is that a number of intracellular organelles or molecules are decided they have to be cleaned up. And what they basically do is they get packaged inside these bodies, and eventually also get phagocytosed. So there's autophagy, actually, in which we've got the autophagosome. So here we see a bit better cartoon of that. So it's in several phases that autophagy occurs. First of all, the so-called cargo recruitment, in which the intracellular cargo recruitment is taking all the bits and pieces that need to be cleared up. These are then given to be enclosed in an autophagosome, and the autophagosome lyses with the lysosome, and the contents of the lysosome spill into the autophagosome and clears up the garbage that's inside the cells. Also not immunogenic. Very nice system. Now, if we look at this particular mechanism, autophagy, in sepsis, for example, we see that the autophagy is severely disturbed. So a normal way of maybe clearing up garbage is disturbed in sepsis, and whereas we have the hyperinflammatory response and the anti-inflammatory response, we have all sorts of abnormal pathways which cause the inhibition of the autophagy. So you've got a lot of garbage just lying around there, not being cleaned up. And in fact, the targeting of autophagy and enhancing autophagy is considered as a potential therapeutic target in the treatment of sepsis. So then we get down to the mitochondria. Of course, the mitochondria is a big issue when it comes to hitting cell death. And what we see here really is the nitric oxides. Here we have peroxynitrate, the different reactive species associated with nitric oxide, reactive oxygen species. All of these things have got a negative effect on the energy transport chain, especially nitric oxide, which has got about a 10,000 times higher affinity for cytochrome 3 than does oxygen. So it directly competes with oxygen in the terminal chain of the complex. And in any case, the result of all of this is the spilling out of cytochrome C. So cytochrome C gets cleaved off from the oxidative phosphorylation, and it spills off, and cytochrome C is a very important instigator for apoptosis, and mitochondrial DNA, which is also cleaved off due to all of these things, then also cause an inflammatory response and results in the clearing up of the mitochondria. So the mitochondria is a specific form of autophagy. And the first thing that happens with a sick mitochondria is the membrane potential of the mitochondria gets decreased, because that's the major functional parameter which drives the mitochondrial chain. And there are all sorts of different pathways which cause that. Now, one of the problems in sepsis regarding the mitochondrial and the clearing up of the mitochondria is that this process of mitophagy is disturbed, and you basically get an accumulation of sick mitochondria inside the cells, not being cleared up by mitophagy, and this contributes, of course, to an abnormally functioning cell. And under normal conditions, autophagy will clear up the mitochondria, so the targeting mitophagy or autophagy can be considered as an important clinical target, and is in fact being looked at into various groups. So each cell, too, has got its own death story. So one of the best ones described, of course, are the erythrocytes. So the erythrocytes also have a suicide program, if you like. It is called Eryptosis, and at a certain point, they too get, if they have come towards the end of their lifespan, or they have been attacked by injury and are not functioning well, they will generate surface receptors, which are then recognized by the phagocytes, the monocytes, and the macrophages, and they will be internalized and taken out. What you can also have, however, is that these erythrocytes become senescent. So they are still there, but they are not really good at transporting the oxygen because of 2,3-DGP, which is not working well, or other issues related to the intracellular homeostasis of the red blood cells. They can disrupt, causing free hemoglobin. So there's a whole story about abnormal functioning red blood cells, and in my opinion, it's one of the easiest ones to follow at the bedside is to follow the last couple of slides I would like to discuss with you regarding cell death is cellular senescence. So cellular senescence really refers to the pathophysiological process by which cells permanently lose their proliferative capacity to regenerate themselves and to become functional again. They, in fact, become zombie cells. So it's been very well described as being one of the key mechanisms in acute kidney injury. So normally speaking, when you have acute kidney injury, excellent paper by Joe Boventra here, acute kidney injury, the tubular cells who form the barrier will get disrupted, cell death will occur, then you will get a reconstitution of the cell barrier because the epithelial cells will allow regeneration, and then finally there will be a regeneration, and so this cell cycle is very important. But what can happen is that this cell cycle process is disturbed, and you have a cell cycle arrest, and what then happens is these cells no longer become epithelial cells, they turn into fibroblasts. And this is the process that results in the fibrolysis of the kidney. And there's in fact a company here, Jean-Louis, I don't know its name, but they have a biomarker which looks at these cell arrest biomarkers as a biomarker for acute kidney injury. So where has this literature been most looked at? It's in the longevity literature. So these are people who want to live forever. And the idea basically is to get these senescent cells and to kill them and to allow, yes, I'm almost finished. And so to stay young, here's a paper in Nature, and boom. So the older you get, the more of these senescent cells that you have. And there are, in fact, now targeted cells, and I think that this is a very promising area for sepsis too. Mostly looked at radiation. This is the FOX04 developed in Rotterdam, and as you can see, a mouse normally looks like this. And if you take away these senescent cells, and what they do actually is they take the senescent cells and make them apoptotic, so they make them suicidal. And that is what I have to tell to you about cell death today. A very complicated subject.